Hello everyone, my name is Rahul, and in this video we will be discussing about basis of classification of animals. So the earliest attempt of classification was made by Aristotle. Aristotle classified animals into two categories, one being called anima and other being called as inanima. Anima includes all those animals who do not possess red blood cells. All animals who do not possess red blood cells are classified under anima, and all those who possess red blood cells are classified under inanima. So all the invertebrates are classified under anima, and all the vertebrates are classified under inanima. Again, inanima is further divided into two categories. One, those who lay eggs, and the other one who give birth to their young ones. Those who lay eggs are called as oviparous, and those who give birth to young ones are known as viviparous. In oviparous, we have examples such as birds, reptiles, amphibians, and in viviparous, we can remember the easiest example, humans or mammals. So Aristotle's classification is no more under use. What we use nowadays is the basis on the level of body organization, body plan, body symmetry, number of germ layers, presence or absence of coelom, presence or absence of notochord, and segmentation. So starting with level of body organization, the hierarchy in biology works as cells make up tissues, tissues combine to form organs, and organs make up organ systems. So this hierarchy is what we have used as a basis of classification. First, we have cellular level of body organization. Cellular level means that body is made up of cells. There are no tissues, there are no organs. So there is no division of labor. Division of labor means that different cells or tissues will perform different functions, but since the body is made up of only cells, each cell will perform all the functions, whether it is respiration, excretion, digestion, etc. So we have example poriferans. Poriferans or phylum Porifera, they have a cellular level of body organization. Next, we have tissue level of body organization. Cells have arranged to form tissues, so there is some division of labor. So there will be some division of labor between various tissues. Example, we have coelentrates. Phylum Coelentrata, they show a tissue level of organization, and phylum Cnidaria, they also show tissue level of body organization. Next, we have organ level of body organization. This is exhibited by platyhelminths. Organ level of body organization is exhibited by platyhelminths. Platyhelminths are also known as roundworms. Platyhelminths are also known as flatworms, not roundworms. Next, we have organ system level of body organization. Organ system level of body organization starts from phylum Nematoda, also known as roundworms, and continues till chordates. What are chordates, what are nematodes, what are platyhelminths, we will discuss later on. The next basis of classification is the body plan. Body plan means how the different structures are arranged inside the body. So first we have cell aggregate type of body plan. Cell aggregate type of body plan is similar to cellular level of body organization. That is, there are no tissues, there are no organs, there are no organ systems. So body is just a loose collection of cells. loose collection of cells, so there will be no division of labor. There will be no division of labor. Example, we already know poriferans. Poriferans exhibit a cell aggregate type of body plan. Next, we have blind sac body plan. Blind sac body plan. Blind sac body plan means there will be a single opening which will function as both mouth and as anus. So a single opening will be present which will function as both mouth and anus. Single opening for mouth and anus. Here we have example platyhelminths. Platyhelminths, they exhibit a blind sac body plan. Also coelentrates, they also exhibit a blind sac body plan. Next type of body plan, we have a tube within tube body plan. Tube within tube body plan. If you have seen a pen looks something like this. There will be an outer tube and there is an inner tube. So the organism is also similar. There is an outer tube and there is an inner tube. So tube within tube body plan. There will be an outer tube, there will be an inner tube. Secondly, there will be two openings. There will be two openings. One will be mouth and one will be anus. Tube within tube body plan will have two openings. One will act as mouth, one will act as anus. Again, tube within tube body plan. This is of two types. First is known as protostomia and second is known as deuterostomia. Proto means first, deutero means second. So when mouth develops first, the body plan will be known as protostomia and the organism will be a protostome. And when anus develops first, it is known as deuterostomia, or the organism will be known as deuterostomic. So in protostomic or protostomia, we have example roundworms, annelids, and arthropods, mollusks. They all have protostomic type of tube within tube body plan, that is mouth develops first. And echinoderms and chordates as well as hemichordates, they show a deuterostomic type of body plan, that is anus develops first, mouth develops later on. Next basis of classification is on the basis of body symmetry. We have two types of organisms or two types of animals. One is asymmetrical and other is symmetrical. Asymmetrical, these animals cannot be divided into two equal halves, by any plane passing through the body. So the body of these organisms cannot be divided into two equal halves by any plane passing through the body. So for example, if an organism has a shape like this, no matter how hard you try, what plane you try, you cannot divide the body into two equal halves. Whereas symmetrical animals are those where body can be divided into two equal halves. Body can be divided into two equal halves by at least one plane. So there may be more than one way of dividing the animal into two equal halves, but there will at least be a one plane that will divide the body into two equal halves. So for example, if an organism looks like this, This plane can divide the organism into two equal halves. This plane can divide, sorry, not this. This plane can divide the organism into two halves. This plane also, this plane also. So a symmetrical animal, the body can be divided into two equal parts, and asymmetrical animal, body cannot be divided into two equal parts. Again, symmetry is of two types. We have radial symmetry and we have bilateral symmetry. In radial symmetry, there will be more than one plane along which the body can be divided into two equal halves. For example, this plane is dividing body into two equal halves. This plane is dividing the body into two equal halves. How? This plane is passing through the middle of the organism. This plane is passing through middle of the organism. The organism here, it is a 3D structure, so the plane will be passing midway. So the organism will be divided into two parts, one upper part and one lower part. However, this vertical plane will divide organism into a left portion and a right portion. So body can be divided into similar parts by more than one plane. More than one plane can divide the body into two equal halves, and only one plane can divide the body into two equal halves. The symmetry is known as bilateral symmetry. This organism, only this plane can divide the organism into identical right and left parts. So example of asymmetrical animals, we have poriferans. Poriferans, they exhibit asymmetry. Radial symmetry, it is seen in coelentrates, and bilateral symmetry, it is seen from platyhelminths onwards. Platyhelminths onwards, we have bilateral symmetry. However, in case of echinoderms, adult echinoderms, they exhibit radial symmetry, whereas larva of echinoderms, it exhibits bilateral symmetry. Next basis of classification is the number of germ layers. So what are germ layers? When an organism is in the uterus, is in the embryonic stage, the cells of the body arrange themselves in layers, and these layers are known as germ layers. Later on, all the tissues, all the organs will be derived from one of these germ layers. So the germ layers are ectoderm, mesoderm, and endoderm. Ectoderm is the outermost layer and endoderm is the innermost layer. In this diagram, you can see this is the outer layer, this is the inner layer, and this is the middle layer, which is not properly formed, this pink colored. However, in this case, you can see this is the outer layer, this is the inner layer, this is the inner layer, and this is the middle layer. Here, this is the outer layer, this is the inner layer, and the middle layer is not properly formed. So when middle layer is not present, the organism will have only two germ layers and the organism will be known as diploblastic. When all the three germ layers are present, the organism will be known as triploblastic. So when mesoderm is absent, there will be only two germ layers, ectoderm and endoderm, and the organism will be known as diploblastic. And when all three germ layers are present, the organism is known as triploblastic. Now, when mesoderm is not properly present, it is sometimes arranged in the form of loose cells known as mesoglia. In diploblastic, we have poriferans. In diploblastic, we have poriferans, and in triploblastic, all organisms starting from platyhelminths onwards, all are triploblastic. So poriferans and coelentrates, they are diploblastic, they only have 2 germ layers, and from poriferans and platyhelminths onwards, they are triploblastic, that is, they have 3 germ layers. Next basis of classification is the presence or absence of a body cavity, which is known as coelom. See, cavity means an empty space. So whenever there is an empty space present inside the body, and that empty space is lined by mesoderm, it will be known as coelom. So whether coelom is present or absent, or there is a false coelom, organisms can be acoelomate, pseudocoelomate, and coelomate or eucoelomate. See in this diagram, this is the innermost layer, this is the outermost layer, or you can say outermost tube, this is the innermost tube, and there is no empty space in between. And here, this is the outer tube, this is the inner tube, and you can see this white empty space between the inner and outer tubes, and this empty space is lined by this pink colored layer, mesoderm. So this is a coelom, this white space is a coelom. Here there is no coelom, and in this diagram there is a white space, but you can see that this pink colored mesoderm is not lining this white space. So such a type of coelom is known as pseudocoelom, when cavity is present but it is not lined by mesoderm, rather mesoderm is present as scattered pouches. So acoelomate means there is no coelom. Coelomate means coelom or cavity is present but not lined by mesoderm, In case of a coelom or true coelom, there will be a cavity lined by mesoderm. So in acoelomate, we have poriferans, coelentrates, and platyhelminths. In pseudocoelomate, we have nematodes or roundworms, and from annelida onwards, we have true coelom. Again, this eucoelom or true coelom is of 2 types. One is known as schizocoelom and the other one is known as enterocoelom. One is known as schizocoelom, other one is known as enterocoelom. Schizocoelom develops as a split in mesodermal sheet. The mesoderm is arranged as a sheet and it splits to form a schizocoelom, whereas enterocoelom develops from embryonic gut mesoderm. What is embryonic gut mesoderm? You do not need to go into the details. We will discuss about this when we will talk about reproduction. So schizocoelom example, it is seen in annelids, arthropods, and mollusks. Annelids, arthropods, and mollusks, whereas enterocoelom is seen in echinoderms and chordates. gynoderms, and chordates. Next basis of classification is the presence or absence of a notochord. Notochord is an elastic rod-like structure, that provides support to the body. So if notochord is absent, the organism or animal will be known as a non-chordate. If notochord is present, the animal will be known as a chordate. So absence of notochord means the animal is a non-chordate. If it is present, it is known as chordate. Poriferans up till hemichordata. All phyla from Poriferans till hemichordata, they are classified as non-chordates, and the other phylum is known as chordates, which includes protochordates or protochordata and vertebrata. What is the difference between chordates and non-chordates? We will deal with this later. And what is the difference between protochordates and vertebrates? We will also deal in the subsequent video sessions. Next basis of classification is segmentation. Segmentation means linear repetition of organs, units, or appendages. Linear repetition of organs, units, or appendages. For example, we have 2 hands, 2 eyes, 2 ears. So this is a type of a repetition. So based on the number of segments, based on the presence of segments in the body, there are 2 types of animals. One who show pseudo segmentation, and others, those who show true segmentation, also known as metamerism. True segmentation is also known as metamerism. Now, what is the difference between pseudo segmentation and true segmentation? If this is an organism, these are the segments. I am numbering this as 1 and numbering this as 5. Again, an organism with segments. This is 1, this is 5. So in case of pseudo segmentation, segment 1 and segment 5 will be of different ages. Segment 1 will be formed first, segment 5 will be formed later on. So segment 1 will be older as compared to segment 5. However, in case of metamerism or true segmentation, segment 1 and segment 5, they are equal in age, that is, they are formed together. So in case of pseudo segmentation, all the segments, they have different ages, and in case of metamerism or true segmentation, all segments, they have the same age. Now, pseudo segmentation is seen in organisms such as tapeworms, and true segmentation, it is seen in organisms such as annelids